McMaster for human parasitology. General information. Preferably, the fecal samples are examined fresh. If samples cannot be examined after collecting them, they should be refrigerated at 4 degrees Celsius. Avoid the use of preservatives such as formalin because these may affect the morphology of the eggs and thus impede a correct diagnosis. The flotation solution should be made at least 24 hours before performing the test and should be stored at room temperature. To ease the examination of a large number of samples and to avoid differences between batches of flotation solutions, it is feasible to make a volume of 5 liters. Equipment lists. To perform a McMaster, the following equipment is necessary. 2 beakers or plastic containers of at least 50 milliliters, a stirring device such as a spatula, a scale, a tea strainer, a Pasteur pipette and a rubber teat, the flotation solution, a measuring cylinder of at least 50 milliliters, a McMaster slide and a compound microscope. Sample preparation. Place an empty beaker on the scale and press the tare key. Add 2 grams of the sample to the beaker. It is important to weigh exactly 2 grams as this may influence the accurate estimation of the fecal egg count. 30 milliliters of the flotation solution is added to the sample. Homogenize the sample using the spatula and pour the fecal suspension 3 times through a tea strainer to withhold the large debris. To obtain optimal results, it is crucial that the debris is firmly squeezed after the sample is sieved for the 3rd time. Once the debris is squeezed to dry, it can be discarded. Filling the slide will be simplified when it is rinsed and tapped on the surfaces. Before filling the slide, be sure that the external surfaces are clean and dry. Place the slide on a flat surface ready for filling. Homogenize the filtrate by pouring it 10 times from one beaker to another. Immediately, place the tip of the pipette in one corner of the chamber and squeeze the pipette until the chamber is full. Do not move the pipette across the opening of the chamber, as this may introduce air into the slide. Rehomogenize the filtrate and repeat the procedure for the 2nd chamber. Examination of the slide. Place the McMaster slide on the microscope table and allow it to stand for 2 minutes. The McMaster slide can be examined using a 100 times magnification. Start with focusing the microscope on one of the grid lines, after which you focus on the air bubbles, which are round in appearance and have a black outer ring and a clear center. Now systematically scan all the columns of both the chambers. Eggs that lie out the chambers are not to be counted. Cleaning of the samples. After the McMaster slide is examined, it can be reused by rinsing it.